we want to provide automatic conversion of data formats in between these tools so that no one has to write a script, and so that people who don't have programmatic expertise are not forced to undergo the effort to get those into a different format. We also want our data in one centralized location so that it can be managed. And we also want any tool that joins to automatically be connected to the entire community of tools. And naturally, we also want it to be easy for new tools to get into the environment. So what are some of the existing ways that people have solved problems like this? We asked ourselves that, and what came up was the use of cloud-based data storage and the use of web-based APIs that have found tremendous success in other places on the internet. So the concept of having cloud-based storage and easy connectivity via web-based APIs was one that we wanted to take as an example in the creation of GenomeSpace. So from these design objectives, we created an architecture, GenomeSpace, and it has three main components. The first component is a set of seed tools, because we wanted there to be automatically a set of tools that people could use that comprised the wealth of functional genomics analyses sort of from the get-go. And also, these also happen to give us a variety of architectures to make sure that we can support all of the different bioinformatics tools depending on how they are architected. We also wanted to support interoperability via this frictionless data transfer between tools. And possibly the most important thing of all, we wanted to make sure that this can support real science. So part of the actual project involves 3 biological projects that will be using GenomeSpace in a essential, basic way to support their research. We want these things to live in the cloud to get all the benefits that you can get from the cloud, basically being the centralized data storage, the ease of transfer from the cloud to other tools, since when you're on the cloud, you're automatically accessible via the internet. And at this point, having established this, we're also involved in an outreach to add new tools to the architecture and to identify new biological projects to expand the scope of GenomeSpace. The institutions that participate represent a wide variety of organizations that are doing genome research from a number of different angles. So to review the principles, we want to aim this towards non-programming users, and we want to support interoperability via having data transfer be something that's done behind the scenes and automatically for you. And the view here is that when you're in Microsoft Word and you want to send your document to someone via email, you don't have to worry about which email program that you're using. Microsoft and your operating system knows that for you. So we said, why can't we do that with bioinformatics tools as well? Also, for those people who know the tools that they're familiar with, we didn't want them to have to change their tools very much in order to participate in GenomeSpace. We wanted this to be minimally invasive and require as little changes as possible to existing tools. So what is sort of behind the hood of GenomeSpace? There is a server component that provides authentication, which manages everyone's GenomeSpace account and also provides a single sign-on for those GenomeSpace tools that are web-based. There is also an analysis and tools manager that manages the tools and their parameters and the analysis they provide. There's also a data manager which manages the repository and all of the file format conversions and makes sure that they're transparent. And these are designed to run in the cloud. And there's also an API component that I mentioned, which is the way that tools communicate with the GenomeSpace server. And this is all in the cloud. So to describe a little bit more about the seed tools and the reason that we chose them, so Cytoscape is the industry standard for genomic and regulatory network analysis and visualization. Galaxy is a tool that is standard for sequence analysis, next generation analysis, and many other pieces of functionality. Gene Pattern is a functional genomics tool that has a large number of gene expression analysis algorithms, plus proteomics, flow cytometry, copy number, and a number of other different tools. Genomica provides the analysis and identification of module maps and co-regulated modules of genes from gene expression data. The IGV is the Integrative Genomics Viewer, and this provides the integrative view of genomic-based information, and any genomic-based information can be visualized and manipulated in IGV. And the UCSC Genome Browser, which is the standard way of viewing genome tracks from all of the currently published and available genome data. So now I'm going to show you live what the GenomeSpace UI looks like and walk through some of its features. So when you first go to GenomeSpace, you're asked to log in, and you can sign in, and as I said, there's an OpenID. So for those of you who are familiar with OpenID, your OpenID account can be used to log into GenomeSpace or any GenomeSpace tool. So, and another thing to observe here is that I have a toolbar that shows me the available GenomeSpace tools. These tools can be launched from the GenomeSpace user interface. And the benefit here is that I don't need to worry about downloading them, registering for them, or anything like that. The GenomeSpace user interface will, if it's a web-based application, will launch it for me, or if it's something like a Java-based desktop application, GenomeSpace will take care of downloading it and launching that for me. You can also see here that there are some additions to the original 6 tools that I just mentioned. We recently announced the addition of Systrome. This is a tool for analyzing ChIP or ChIP-seq data, and these analyses will identify areas of the genome that bind proteins, and you can use them to identify promoter regions, enhancers, repressors, and other controllers of DNA replication. And here on the right hand are the data sources. And in addition to the UCSC table browser, you'll see InSilicoDB. And InSilicoDB is a very nice and fully featured interface on top of the Gene Expression Omnibus, GEO, which allows you to navigate and export GEO data sets in some very powerful ways. So below the toolbar, you'll see my cloud-based file system. And this looks just like a regular file system that you might see in Windows or the Mac or Google Docs. And I can navigate through my files, and I can do various things with files that you would expect. One other thing that I can do in GenomeSpace is that I can share my files with other users. I can set up groups, and I can specify, for example, that my AML/ALL directory can be shared, and I can either select a group to share this with, or I can make something publicly available. This is useful for when someone does an item of research with GenomeSpace, and they want to make the result data sets available more widely. You can create a URL that will link to a result data set within GenomeSpace, and you can publicize that just like any other internet-based URL. So now, how does GenomeSpace look to the other tools, and what's involved in connecting to GenomeSpace if you are a tool? For this, I'm going to go back to the slides and show you what GenomeSpace looks like in some Java-based tools and some web-based tools. The first one is IGV. So I have a separate menu item in IGV for GenomeSpace that will allow me to load data sets from GenomeSpace and save sessions to GenomeSpace. And the way that I would interact with that is to click on that, and you can now see that I'm seeing a representation of the data that I have stored in my GenomeSpace account, but this is coming up in IGV. And so I can navigate through this, and I can select a data set, and I can click Open, and I'm now viewing the contents of that data set in IGV. And what I've just done is I've taken my cloud-based data set, and I have automatically loaded it, and I'm visualizing it in IGV. How would this look like for a web-based tool? So let's take a look at Gene Pattern. So you can see that I have a special login on the right-hand side for GenomeSpace. This accepts my OpenID, and I click Sign In, and you can see here that I have now a new GenomeSpace tab. So this starts to give you an idea as to how you can, how tool developers can customize GenomeSpace for their particular tool. I also have a view of my data sets, again here in my GenomeSpace cloud-based data, and this allows me to send this data to any gene pattern analyses in a way that is sort of indigenous to the way that gene pattern does it. Another key piece of functionality is the ability to do automatic data conversion on the fly, as I mentioned. And that's illustrated in this section here, where I have the opportunity to send to a number of these other GenomeSpace tools, and I'm given the opportunity to also automatically translate these file formats to formats that these tools accept. So for example, I can send this to Genomica as a GXP file, even though the file itself is not a GXP file in GenomeSpace, and the GenomeSpace will handle the translation behind the scenes. Now, what does GenomeSpace have to offer data sources? We're taking a look here at InSilicoDB, and InSilicoDB provides a number of different opportunities for login, and there is a GenomeSpace-specific OpenID login, and once we've logged in with our GenomeSpace account, we see a number of different gene expression data sets. When we've selected one and we want to export it to GenomeSpace, we select Export, and we can see that GenomeSpace has been selected, and once Export is clicked, this data set will automatically be created by InSilicoDB and transferred to my GenomeSpace cloud-based data storage. So we are right now in the process of enabling a number of additional tools. Reactome is a database of protein-protein interaction networks. GE Workbench is a network analysis tool that allows you to impute networks from gene expression data. Taverna is a web-based workflow tool. ArrayExpress is an additional repository for gene expression data, one of the industry standards. And the National Center for Biomedical Ontology provides ontologies that will allow us to help describe all these tools, and for when we have a large number of tools in the system, to create tags and vocabularies that will allow people to search very easily for these tools. So let's take a look at how GenomeSpace can be used to support real science. And for this, just because of the nature of the analyses that I'll be taking you through, I'm going to be doing this largely slide-based. This can all be done within GenomeSpace, but just because of the time it takes to do the individual analysis steps, I'm going to use slides. So an overview of what we're going to be going through is here. We're going to study the regulatory control of human hematopoiesis. And what we're looking at here is an analysis step view of each of the parts of research that went into this particular publication. And the part that we'll be showing in GenomeSpace is part 5. So to focus down on this, what we'll be seeing is the loading of an expression data set into Genomica, and that's going to determine our transcriptional factor data sets. This is going to be, the data set itself is going to contain about 200 samples and 8,000 genes, with also a database of transcription factors. We're going to take that data set of transcription factors, and then we're going to go into gene pattern, and we're going to run a differential analysis. That's going to tell us which transcription factors are active between the two conditions that we're measuring. We are then going to send that data back to Genomica, and we're going to run a module networks analysis to get a list of the regulators of those transcription factors that we determine to be the significant ones in hematopoiesis. And we are then going to view annotation tracks of the transcription factors compared with known SNPs and linkage regions in those genomic areas where the transcription factors occur. So the first thing that we do is that we go into Genomica, and we create our transcription factor data set. This is what I see when I launch Genomica for the first time, and you can see that GenomeSpace will download this software for me. So I then see my Genomica screen, and it's empty, so I go into GenomeSpace, and I choose to load my data from GenomeSpace, and we're looking at the expression data here in the main section, and all of these little blue dots correspond to transcription factors. And I want only the genes that are these transcription factors. So I export that to my GenomeSpace data storage. And I do that from the File menu. This is how Genomica does it, and I choose Save to GenomeSpace. And I now see that my files have been saved to GenomeSpace when I'm back in the GenomeSpace user interface. So what I then want to do is that I want to send these files to GenePattern. So the first thing I do is I select them, and I click Launch GenePattern. And GenePattern will automatically launch with those two files that I selected. And when I see GenePattern open, it will show me that GenomeSpace has sent me a file, and I can then save those files to my GenePattern analysis area. And what I then do is in GenePattern, I do the analysis called comparative marker selection. This is going to indicate our differentially expressed transcription factors. I run this analysis, and I then send the results to Genomica as a GXP file. So this is one of the most powerful functions of GenomeSpace, because this is exactly where we do not need to write any scripts to convert from one tool to another or one format to another. We can just send data directly to the tool that we're interested in. And Genomica here opens with just my expression data set of transcription factors. And when I'm here, I run my module networks analysis on the expression data, while also loading the lineage-specific transcription factors that we just generated into GenePattern. So what this allows us to do is to browse through our results, and we can see a module that has as the top regulator SMAD4. And I don't know if that's visible from this slide, but what this does is it divides our sample space into two partitions, while the other regulators further subdivide these samples. So we see that the result that we get from our module networks analysis agrees with the result that we get from our differential analysis, in the sense that both are coming up with significant results. So we want to take the next step, and we want to visualize what these transcription factors look like on the genome. So for that, we go to the Integrative Genomics viewer. And so bear with me for a second. My slides are not advancing. Okay. So I now select the genomic coordinates here, and I add two genome tracks that contain known SNPs and transcription factors, and also the RefSeq genes for transcription factors. And I then click to launch these in IGV. And what I now do is I can view the genes that I have determined to be significantly differentially expressed transcription factors, along with known SNPs and linkage regions, so that I can see where they line up on the genome. Additionally, since Galaxy is also in our tools, I can actually compute a measure of the overlap between the transcription factors and the tracks that I have just added. So this is a demonstration of what's possible when you can put all of the genomic analysis tools at your fingertips and remove the friction that's involved in transferring data between one tool and another tool. So where we're at right now is we have several hundred users of GenomeSpace. As Kevin said, we announced in late April, GenomeSpace is an official public beta, and we are looking for researchers who have biological projects that they're interested in using GenomeSpace to support. We are also looking for developers who want to add their tools to GenomeSpace and contribute format converters and help us with the infrastructure. And I should add that GenomeSpace is freely available, and it is open source using an OSI approved license. So it is a completely open source project. We're also interested in hooking up with data providers who are interested in making their data sources more widely accessible and easily translatable to GenomeSpace tools. So with that, I wanted to acknowledge the people who are responsible for creating the vast number of moving parts that go into GenomeSpace, the collaborators, which include the development teams of the seed tools, as well as the researchers on the driving biological projects, and the developers of the infrastructure at the Broad, as well as our funders, NHGRI and Amazon Web Services. And as I mentioned, we're very interested in reaching out to people who are doing this research and interested in what GenomeSpace has to offer them in terms of connection with existing bioinformatics tools. And at this point, I'd like to thank Kevin and Janine again and see if there are any questions.
Michael, thank you very much. Kevin Davis back with you all. Fantastic presentation and live demonstration. And we are now happy to take questions from the audience. We have plenty of time left within the hour to do so. You may not have seen the chat box because we're in desktop sharing mode, but if you hover your mouse it's at the top of the screen where you see the little green bar that says Viewing Michael Reich's Desktop. That will bring down the chat box, and you can enter a question there, and I urge you to do that right now. You may have some technical questions or more specific scientific questions, and I'd love to have Michael address those if we can get to them. So do get typing away. In the meantime, Michael, I've got a few questions just to get the conversation going for our 170-some participants on the webinar today. You're part of, in the introduction I mentioned, a sort of a cancer informatics group, but this is much more than a cancer-specific tool, I take it.
Yes, there is nothing special about any of the tools that are in GenomeSpace right now. Any tool can join, and it doesn't need to be cancer-related. It is meant to be generalizable to all bioinformatics tools, and indeed could be used for non-bioinformatics tools as well. It's really a connection layer in between tools.
I think most, if not all, of the tools that you mentioned have emerged from the academic community. They're open source or what have you. Can GenomeSpace work, or is it compatible with commercial tools? What if I'm trying to integrate software from, I don't know, an Ingenuity or an Acceleris or a CLC Bio? Is there a way I can do that? We are right now speaking with a couple of commercial organizations who are interested in using GenomeSpace and making their tools available that way. And it is very possible for any commercial organization to connect to the GenomeSpace server and use it as a data source. And right now we're working through the issues of things like displaying a commercial tool in the tools menu. We want to make sure that the people who are able to use that are the actual customers and things along those lines. But we are certainly pursuing collaborations and interaction with commercial organizations and their products. What if I'm a fan of Ensemble rather than the UC Genome Browser? Right now we are reaching out to a number of different organizations, and we're in differing stages of communication with them. So I'm hoping that we will be able to get the lion's share of genomics analysis tools incorporated into GenomeSpace. And there are different ways that different tools can participate. For example, tools don't need to be
fully 100% GenomeSpace compliant in terms of translating their data sources and things along those lines. GenomeSpace tools could just send their data to cloud-based GenomeSpace, and this is a sort of easy way for tools to participate. So for those people who are developing tools, there are various stages of GenomeSpace enablement, and it is very easy for people to get in on sort of the ground floor. So that's something that is available to just about any web-based genomics tool, and we expect to be announcing more in the near future.
Excellent. We have a flurry of questions for the audience, which I will get to in just one second. My final question for now, Michael, however, is in the early part of your presentation, you talked about the half dozen seed tools that you've used, and you also briefly mentioned, I think, a trio of biological tools, and I think I noted them down as cancer stem cells, link RNAs, long non-coding RNAs, and patient stratification. I wonder if you could say a little bit more about what those tool sets consist of, and are they designed to be a way that people can kind of kick the tires of GenomeSpace and yeah, without having to upload their own data sets?
Sure. Those are research projects, and we are supporting those research projects through the GenomeSpace tools. So the idea behind that is that in the course of doing this research, the researchers will require various tools and analyses. So we will make sure that GenomeSpace has the capabilities to support that research. So they're not actual tool sets themselves, but they're going to produce workflows between those tools that we are going to publish and that people will be able to use in their own research. And I also wanted to bring up the tools that are available on the GenomeSpace website for people who want to get involved. We make it very easy for people to learn the basics of GenomeSpace because we understand that not many people know every single tool in GenomeSpace. So what we've done is we've provided a list of what we call recipes that will teach you just enough GenomeSpace in order to use the tools that you may be interested in. And these are separated into how to load data into GenomeSpace from various tools, how to move data between tools using GenomeSpace, and how to analyze data in GenomeSpace. And when I click on this, it will give me a step-by-step tutorial as to how I can use GenomeSpace to do these fairly general analyses. And so these are sort of short versions of the biological workflows that we're creating using those larger biological projects. So I would recommend to people, if they want to understand how to use GenomeSpace and a little bit more hands-on what it's about, to go through those recipes, and that will give them a feel for how to use GenomeSpace.
Excellent, Michael. Okay, we're going to now go through as many of the questions as we have from the audience. They're still coming in. We've got about 15 minutes left in the webinar, so we'll try to keep the questions and the answers fairly succinct, but obviously I'm sure Michael won't object at all. If anyone needs further information, I'm sure he can devote a little bit of his free time to handling some questions offline if we don't provide quite the amount of detail that you're looking for or are unable to get to your question because of time. So from the top, Michael, you may have addressed this already, can you install a Cytoscape plugin into a running GenomeSpace instance?
GenomeSpace has its own version of each tool, and so the way that we handle requests for Cytoscape plugins is that we add those plugins to the GenomeSpace version of Cytoscape, and those plugins are then available to anyone who launches Cytoscape from GenomeSpace. So the specific answer to your question is it's a little bit different from the standard way of doing it in Cytoscape, but we're glad to work with people who want specific plugins. And the eventual view is that you will be able to do that. You will be able to add plugins to your own version of Cytoscape within GenomeSpace. And this is also a good opportunity for me to say that one of the things that we are working on in the in the recent future is going to be the ability to have your own private version of tools that maybe are not on the official GenomeSpace server, but that you can launch from GenomeSpace and manipulate that way.
Next question, two-part question. Is there any size limit for the input file, and do you have any plans to enable to apply this to genomes other than humans?
Right now, the only size limit is the limit of the cloud-based data storage that people have in their accounts. And right now, because we're in beta, we don't have a hard and fast limit on that. We're really just observing the usage patterns right now. So that the answer to that is no. And in terms of which genomes we make available, it is not just specific to human genomes. We have a publicly shared area in GenomeSpace that provides data sets for a wide number of different organisms. So the analyses themselves and the data are not specific to any particular organism or genome.
The next question, is there a programming interface instead of the user GUI to integrate between the different tools using this framework?
Yes, there absolutely is a programmatic interface. We provide a web-based API that will allow you to do all of the things that I demonstrated that you can do in an actual tool. It will allow you to connect, to transfer data, to bring in data, and to transform data from one format to another. So absolutely that's available. It's available as an API, also as a Java package, and it's also been imported to Clojure for those people who are interested in that.
Curtis wants to know, there are a lot of analyses involving very large files, FASTQ, BAM, and so on. What are the limits of your cloud storage? How does billing happen for that? Aren't the data transfer times a huge problem?
So data transfer times are a large part of analysis of NGS data. And what we're doing in terms, first, of the storage is, as I mentioned before, we're observing the usage patterns to determine what sort of size policy we're going to apply toward the cloud-based storage. But another feature of GenomeSpace that is going to come up within the next couple of months is the ability to connect GenomeSpace to other types of cloud-based storage. For example, if people have their own Amazon account, you will be able to link that to your GenomeSpace storage. And we're also looking into other types of cloud-based storage, such as Dropbox and Google Drive, to allow us to connect to the widest variety of data sources. In terms of data transfer, what we're trying to achieve there is to, in keeping the data in the cloud, we're putting the data closer to where it's being analyzed. So that will realize somewhat of an increase in analysis time. But we're also looking at methods for analyzing data in smaller chunks and essentially streaming pieces of data such as FASTQ and BAM files so that the analyses can be done before the files are actually uploaded, so that can be done in a more pipelined way.
Thank you. What types of data, is the next question, can be input into GenomeSpace? Is it limited to just expression, RNA-seq, array-CGH, DNA-seq? Are there other types of data perhaps we haven't talked about that you can incorporate as well?
Yes, there are no limits to the type of data that can be added to GenomeSpace. The only limits are the types of data that all of the tools will accept. So if a tool will accept it, you can upload it in GenomeSpace. And even if not, I probably shouldn't say that you can store your Microsoft Word or PDFs in GenomeSpace storage, but there's no requirement that files be in a certain format to be stored or used in GenomeSpace.
We've already touched on the integration of commercial tools. I'm just curious, Michael, I mean, if there are, and I know there are people from industry on the webinar today, if they were interested in talking to you more about GenomeSpace, can they contact you directly?
Yes, we'd be glad to speak with anyone from industry.
That's what I thought you were going. That's what I thought you were going to say. Is there any, next question, is there any local caching of the data that is cloud-based?
The data that is cloud-based is not cached locally. the way that we deal with, and I'm going to make an assumption here that the person who asked this question is wondering about how to deal with the fact that there are certain tools that you can run on the desktop, so it's inefficient to have to upload a file to cloud-based storage just to download it to your desktop to have it analyzed. So the answer to that is that it is possible in GenomeSpace to send a data set from your local storage to a GenomeSpace tool directly. It is possible to bypass the cloud-based storage. So in that sense, we don't specifically cache it, but we get around that problem in a different way.
All right, we have about 10 more minutes left. We have plenty of questions to occupy us for that time. Hope you can stay with us, talking to Michael Reich from the Broad about GenomeSpace. Michael, quick question. Can I launch a large BLAST job on GenomeSpace?
Yes, if someone were to want to add a BLAST server to GenomeSpace, right now we don't have tools that run BLAST, which is the only reason for that is that we don't have analyses that require the use of BLAST. But if we were to add a tool that runs BLAST, there would be no problem in launching a BLAST job from GenomeSpace.
Is it possible to install GenomeSpace in a private cloud, and how much effort would be required for installing and maintaining it? How large is your current Amazon infrastructure? We have gotten that question a fair amount, and right now GenomeSpace has been designed to run on the Amazon cloud. We do have, as part of the long-range vision, to be able to make it sort of transportable to other types of cloud infrastructures. But right now, the next step that we're taking along those lines is to allow other people to launch their own GenomeSpaces on the Amazon cloud, and that's something that's going to be coming up in the, I'm going to say near future, but I can't put a specific date on it. So we would like to make this available on private clouds, and right now we're weighing that against the other development requests we have. Do you want to quickly say anything about how the size of your current Amazon infrastructure?
I would say it is, the server itself is very, very manageable. While it supports a large number of tools and analyses, the server infrastructure itself is, unfortunately, I couldn't tell you how many lines of code it is, but it's along the lines, if those people are familiar with any medium to largish size software project, it is something that has its own build system that people can import and use it to build their own GenomeSpace. So I guess by way of answer, I would say it's not so big that it can't be built using the standard tools that software development best practices are currently using. And that's, I'm sorry that I can't give you a more accurate assessment.
No, let's press on. In terms of pathway analysis, how does GenomeSpace compare to Ingenuity or GeneGo, for example? So there are a number of different pathway analysis algorithms that span the different tools within GenomeSpace. The GE Workbench, for example, provides the reverse engineering of networks based on gene expression data. Cytoscape, which is the main tool that we promote for pathway analysis, provides a number of different analyses and connections via its plugins to Go and to a number of different protein-protein interaction networks. Gene Pattern as well has a number of, for example, gene set enrichment analysis, which also identifies gene sets that are significantly differentially expressed. So in terms of how these tools compare with the commercial tools, I would say the main important thing is that if people are interested in integrating commercial tools into GenomeSpace, we are interested in exploring that. The functionality is fairly similar, and these things tend to depend on the specific details of what research you're working on. So we need to kind of get a little bit into the research in terms of which one would be best for you. But I would say that the functionality is quite comparable to a GeneGo or Ingenuity. All right, last 10 minutes now, so nice crisp answers, Michael, if I can trouble you, so we can try to get through as many of these as we can. For data source integration within InSilicoDB is the next question. Am I downloading data from Geo to my Amazon cloud space, or how exactly do I retrieve and store the data?
You would go into InSilicoDB, you would click Export, and that would handle the transfer to your GenomeSpace data storage. So that would be done for you. You don't need to do anything in Geo.
Can I integrate non-commercial software running from my desktop with GenomeSpace?
We are working on the ability to do that, and so we expect to have that available over the next several months.
Will GenomeSpace be free forever? Who will pay for storage and CPU time? GenomeSpace will be free for the length of time that it exists. I think that I can say that. We may need to change at some point the data storage policy, but the use of GenomeSpace will be free. Plans to make GATK part of GenomeSpace?
Yes, and I should say that there are all, through the tools that are in GenePattern and Galaxy, there are already a number of GATK-based tools already in GenomeSpace. There are variant callers, RNA-seq QC analyses, and a number of other GATK-derived tools already in GenomeSpace.
A specific question or case study from Rick. I wonder how GenomeSpace will work against the rock of big data, he asks. Say I have 200 million bases in 10 different RNA-seq samples. I want to do a de novo transcriptome project. How well does this work? Where does this massive amount of data live? You're chuckling already, Michael. Where does the computation take place if I'm using Trinity to do the first pass assembly? The program takes a lot of resources.
Yeah, that's an excellent corner case to ask about. And what I would say, the important things to keep in mind are GenomeSpace does not dictate where you are doing the analysis. So, for example, when I was demonstrating the GenePattern and Galaxy analyses, the GenePattern server was running on the Amazon cloud. The Galaxy server was running at Penn State, where Galaxy is developed. So there are no preconceptions about where you're doing this analysis. So you could, in fact, have a massive compute node that's running Trinity somewhere that you could farm that job to via one of the GenomeSpace tools. For example, putting it into a private gene pattern. As far as the huge data sets, yes, right now we would have those data transfer and storage challenges, and as I mentioned, we're working on those because we know that the conditions that he mentioned are going to come up probably more and more.
What is your strategy for using tools that have not been specifically modified for inclusion in the GenomeSpace ecosystem? So right now there is a very sort of modest minimum that is required for adding tools to the GenomeSpace. We are right now working on making it more easy to launch generic tools in GenomeSpace so that all you need to do is put them in your control panel so that they can be launched. That's sort of a stage 0 GenomeSpace enablement, and we hope to have that available in the near future. But that corresponds to sort of, what it means to have a tool that's not in GenomeSpace yet launchable from GenomeSpace. Is it possible to customize GenomeSpace by incorporating our local version of Galaxy and its associated tools, asks Sean.
Yes, it is, and in fact, the new tool that we added, Systrome, is in fact a Galaxy server that's been repurposed for different type of analyses. So when we announce the availability to run private tools, you will be able to have those capabilities automatically.
What are your plans for training, on-site or off-site training or workshops?
Right now, we're designing a series of workshops that we're still figuring out the schedule for, but between now and then, we're creating a number of of screencasts that will show people the features of GenomeSpace in depth, and we're always adding to the documentation that's on the genomespace.org website. So between those three things, we expect to be able to really keep people informed about how to use GenomeSpace. We're also going to be at the community conferences for a number of the GenomeSpace tools. In July, we'll be at the Galaxy Community Conference, and we plan to be at the Cytoscape Community Conference in December, and we'll be doing workshops there. We'll also be at ISMB this year in Long Beach.
I'm just going to quickly pick a couple of other questions. What about integration with Illumina Backspace? Is that planned or has it begun?
I'm sorry, but I'm not familiar with Illumina Backspace. So I'm
is
sorry,
that for the MySeq? Okay.
Oh, yeah.
BaseSpace. Excuse me, I misread. BaseSpace.
Oh, BaseSpace. Yes. So I should also say, in terms of integration, there's a number of different questions about integration. The API that we've created is extremely accessible, and it makes it very easy to integrate with any number of these tools, regardless of the development environment that they were used in, and that also applies to BaseSpace. So it's a matter of making the connections with these organizations and getting those into GenomeSpace more than the technical requirements themselves.
Any browser compatibility issues, Michael?
I would recommend that people use Firefox, Chrome, and Safari. We found a few issues with IE9, so if people can avoid that at all, that they would get their best experience using something else.
And maybe we'll just take this final question. I wonder if you may have tackled this already, but can you say whether you can get the source code and run these tools using the GenomeSpace platform, but storing the data locally, not in the Amazon cloud?
So the first part of the question, the source code is available. It's hosted on Bitbucket. So anyone can go to Bitbucket, get an account, and fork the GenomeSpace project. So you can do that. In terms of running these analyses, those are issues involving the individual tools themselves. So these tools can be and are designed to be installed and run locally. So you could do all those things, but what you would need to have to put them all together is sort of your private GenomeSpace. So that's something that I would encourage people to wait on, because we'll be developing that shortly.
All right. Well, Michael, we've kept you busy for the last 60 minutes, and I think we probably should draw proceedings to a close. But thank you so much for a fascinating presentation. And thanks to everyone for staying on the call. We had nearly 200 people at the peak of the presentation, and we've received dozens of questions illustrating the intense community interest in this tool, and I'm sure people will want to follow up with you offline if we were unable to get to the question, or you had to give a very succinct answer in the interest of time. You can reach Michael via email, mreich@broadinstitute.org, and find more information, of course, at www.genomespace.org. It's been a privilege on behalf of NGS Leaders to host this webinar and to provide information and an overview about a tool that very much fits the mantra of NGS Leaders, which is to provide resources and for the NGS community, and provide tools that help and information that help expand your research and make it more effective and more efficient. So thanks to Michael and his team for all the great work they do at the Broad, and we look forward to following the evolution of GenomeSpace in future webinars, I trust. We have a survey for people to complete. I don't know if we can put the link up on the screen. If we can't, we will email it to you, and I think it's appearing probably right now. If we we're unable to show you the link, we will email it to you just to get your take on the webinar, as well as your input on the community and topics for further webinars would also, of course, be very much appreciated. That's in large part how some of our most recent and successful webinars have come about. It's providing information that you want to hear, so that's what we're here for. So on behalf of all of my colleagues, and in particular, I just want to thanks once again Michael Reich from the Broad Institute. Michael, thanks so much for all of your time. I hope you enjoyed it.
Thank you, and as Kevin said, I'll be glad to answer any questions if people have more technical things that we couldn't get to in this talk.
That's fantastic. I think we all want to, on behalf of everyone, extend our thanks for your time and work on behalf of the community. So on behalf of my colleagues who put this webinar together, I'm Kevin Davis for BioIT World and NGS Leaders. Thanks for joining us. Please visit the website, and we hope we will have another exciting webinar for you in the very near future. But for now, thanks for listening. Thanks for viewing. Take care. Bye-bye.